Check, 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 check. Check, 1, 2, 1, 2.
Check, check, check.
I hear you. Sending you content. Cool. Okay. Thanks. Bye. Check, check.
Right. It just makes it hard. They're freaking out. They're going, oh my God, do we have somebody to hook up the equipment? RSVP, or they invited a bunch more people than RSVP, and I'm like, I only ordered 20 lunches. Y'all need to figure that part out. Maybe you're gonna have a bigger box. Yeah, go ahead and take that one in.
That's how you read. Have a good day. You too. Thank you. I just need some patience. Just do that thing. See you later.
Okay.
Yeah, that should work.
Karen? Yes. That's a greeting. It's over with.
Oh, nice to meet you. I'm Allison. I'm the
One person was, but
coordinating our seminars.
Oh, thank you.
Need help getting your presentation set up?
I'm just turning it on.
Okay. Do you have any videos in your presentation?
no.
Okay. There's not for your computer.
So the mouse just controls the arrow.
Yep.
Yeah.
Yeah, right and left for forward and back.
But I would
This is your mic.
And we need the, yeah, it's up there. So, see my 4 dogs. 1, 2,
Oh.
big one. So, yeah, when I moved to Atlanta, I ended up with 4 dogs and a cat.
Wow.
Yeah, that was an adventure moving them.
So how do you travel?
Well, I'm down to 2 dogs and a cat.
Okay.
And now I have a friend, one of my friends from graduate school. If it's like 1 or 2 nights, she doesn't mind staying. And if it's longer than that, I board them.
Yeah.
But when I very first got there, I had to board them all the time.
Yeah. For like 3 weeks. I'm like, person.
Bar. Presentations there.
I'm gonna go over there too.
Oh, this always happens to me. I don't know why it does this.
So this is for people who want to be able to see like what's coming on deck and like extra notes. It's basically just separated
Okay.
your computer into dual displays instead of mirroring it. Do you want it mirrored instead? Okay.
Can you make that a permanent change?
Yeah.
Why is it so interesting?
Yeah, I think you hit this. This is what you hit. There you go. Yep. Welcome.
Really quick, make sure it's doing what it's supposed to do. Because sometimes things don't. Okay.
But then it's already like advanced and stuff. Yeah. And trying to figure out who she is. I got it. But she's so. Sorry about that.
Yeah, they have a consortium that are
And then I'm thinking longitudinal, we can wait till like Monday or Tuesday.
Let's do on Wednesday, right?
Yeah. Because I'll probably need the weekend to actually be able to work on it.
Too.
So maybe Monday or Tuesday we should.
you get the older around the front and then come
That sounds like a good plan. I have so much to do. I didn't do anything.
I do.
I'm still overwhelmed.
I did things, but I'm still feeling overwhelmed. Next week, I'm teaching a class for David.
Oh.
Your face is spending a lot of time at that cabin of yours.
It is 6602. Hi,
Oh,
how are you?
Good, thank
Oh
you
my
for your
gosh.
analyses, like all your
Yeah.
help with all of it. It
No
actually was
problem.
great.
Okay, good. Yeah, good to see you.
We're still going. Everything is
Okay.
still like rolling out.
Yeah.
We might make like 100 changes. Oh, it was
Yeah.
really helpful.
Yeah, good. I'm glad. I wasn't sure if I met all of your
I was
questions.
all over the place.
So,
Really was.
so I just sent you everything that I thought. So,
Great. So thank you.
yeah, no problem.
Cool. I'm gonna go talk to my program director.
Okay.
And
I thought you were talking about relativity.
Yes, but the whole thing,
Gravity and how that bends time and space.
like Interstellar. I'm like, whoa. Did you see that movie?
Yes,
with,
I
I know. Yeah.
It's there, but it's not there, but it's a day here, and it's
Ready. But
Wait,
I,
last week?
Yeah.
How'd it go?
You know, it's fun. Genomics too?
So as
It wasn't
icebergs
really
or
straight.
something, and
Yeah.
they melted.
Comets are basically huge icebergs.
So why would some be fresh and some be sea?
Why would what?
Some be fresh water and some be salt water.
Oh,
which means this week is
I
off.
feel like they should at least break it up between.
I finished everything before I left, but I didn't do any thesis stuff because that was like where the
Well, the freshwater, I think, is just the process of evaporation.
it's just been frustrating because we thought we had a plan, and then I started looking at the data and summarizing things, and
Well, probably not.
and the plan is just completely.
Your presentation today, then. So don't feel compelled to provide those answers if you don't feel even know.
Please.
You're going to be up there and
No, I will just call on Tim.
That's what I experienced.
Talk to you about things beyond this.
But back to the comment thing.
actually, Anna looked at
5 years on Yeah. the longitudinal thing that we're interested in. Yeah. But then the interval centered is we only have measurements at 0, 3, 6 months.
yeah, which isn't that many.
So you basically can't use hardly any of the longitudinal things.
But Miles is going to get you
You can't use any of the longitudinal measurements within 3 to 6.
yeah.
Because you don't know which came first.
Right. So I'll pick you up at the next from Miles at about 2:15 to 20 and take you to
The month if the event happened at 5.5 months.
then
I don't know. It's
how,
just all
you don't have any data.
Well, yeah, so he's telling me about all these things he's interested in, and you didn't record enough information for that.
Right.
So it might end up being I do something that's not really relevant to the doctor, and that's. Anyway. You were at USC. You worked with Chris.
Yeah.
Fred left.
So,
Question or anything.
Complicated.
Every week, and it's we've come up with anything new since last time.
I wish I left an open senior faculty tenure spot. And
Right. Yeah. Just pull it from.
oh, sad to see you go, but yeah, it'll be interesting to see what he builds with that because there's plenty of, which is good because they needed to build something new.
Right. And so then she's asking me questions well, we don't know this. I'm like, well,
You do.
I was reading that was pretty bad.
Okay, perfect.
I know he told me, but I don't remember.
and I'm somewhere in between. Yeah.
Pretty much about what his plans were.
When, Lauren sends me wants to come to this talk. And actually, I'm helping organize it. Such a small, again.
Be careful, you were never here.
That wouldn't be nice to everyone.
Yeah.
Is everything going okay?
I am really excited to hear about your students at USC, and
Yeah, I'm trying to figure out where they went.
they already left.
No, they were.
So we organized a room for them so that they can use our Zoom thing. My student will definitely be there. One of my other students is actually...
Right, because I picked each space on shift, but I didn't see their eyes.
Yeah.
But I didn't.
So yeah, I was going
Still trying to figure out students here. There's
email everyone again
She did? When?
last week.
Water and then microwave?
no one. Really want to do cancer. I have on whose committee I'm on that's looking at influenza, and I'm like, well, that could be cancer somehow, right? We could do something cancery with that. I'm pretty sure.
It tastes pretty good. Yeah. Epi.
I'm Emily. Whenever you.
On an ASPO call recently, we started talking about, I feel like the number of graduates, PhD students, finishing their doctorates in cancer epi specifically is going down.
That's quite a big program. I didn't think that we did, but I think we probably had a fairly large program. So at the moment, in their second or third year, we have
At a brewery
psychic, so I sort of assumed that.
How are you? So I called them and they have it.
Oh, I tried. I'll be right back.
But they're like out camping, like in the national park. So they don't know, but I like left my mom like voicemail, text, like go here, call me when you get service.
Yeah. Most of the time it's behind some.
I know. They're coming? No. So do you live
like when you go in.
Ben Stiller, what came out last week, it's a lot of
the whole Ben Stiller thing. Oh my god. The drama behind the Ben Stiller.
The whole what?
The whole drama behind
Lori Crane used to do. She did a develop an intervention.
What's the latest drama?
So something about
she was, but I don't know if she's kept up on it since it's been recommended.
Otis and Rich, of course, are disagreeing
on the process to
I work in the School of Pharmacy, toxicology.
Review paper.
pharmacy, the department of college or department.
No, this is a separate building. This is near the lab.
Oh my god, it was like the, classic like butcher paper. Yeah. Here's a tray.
Okay, what do you want?
Oh, you can have
I don't know why you're asking about my mom. Well, my mom. Let's just talk about my mom, actually. Doing really well.
Yeah. Oh, coma. Okay, okay. Okay, next time.
I'm going to send you an email. Do you have? Oh, I have two. I have two. My daughter's outside on
Cancer Center reviews. I've been on the parent committee. First couples costume. I'm so excited. Oh wow. Okay. So yeah.
Almost done, yeah, okay.
I joined as a postdoc.
All I do is reporting.
And then just
Research
Hi, everyone. Is this working? You hear me? Not that you really need it in this small of a room, but my name is Betsy Risendahl. I'm an associate professor in community and behavioral health in the Colorado School of Public Health. And thank you for coming today to this special seminar that's jointly sponsored by the Department of Epidemiology in the Colorado School of Public Health and the University of Colorado Cancer Center. And it's my pleasure to have our esteemed guest, Dr. Susan Gapster, who I've known for 20 years. We first met at the University of Arizona in Tucson when I was a student, and we were working together in cancer prevention and control. Susan has since gone and worked for 15 years, is that right, at Northwestern, and about 8 years ago transitioned to the American Cancer Society, where she's the Vice President of Epidemiologic Research, and she'll be talking with you about a lot of her work today. So thank you so much, Dr. Gapster.
Thank you.
So my talk today isn't going to focus just on my work. And in fact, one of the reasons I went to the American Cancer Society is because they have these very, very large, huge studies, and for an epidemiologist, it's literally like being in a candy store. You don't know what to do first, you don't know where to turn to first. And so what I want to leave you with is a little bit of a sense of the history of cancer epidemiology research at the American Cancer Society, and really the impact of the research studies and how we think today about some of the research we're doing. I also will talk a little bit about what I think are some amazing collaborations that really allow us in our program to extend our expertise. And towards the end, I'm going to do a little bit of a plug for a new program that we've just developed that I'm really excited about. And hopefully, if anyone has any questions at the end, I'm more than happy to answer them. So I want to start off with the mission of the American Cancer Society, and this is actually a little bit old. I haven't updated it yet. It's been tweaked just a tiny bit recently, and it's actually not a nationwide anymore, but we are now a global community-based voluntary organization. But I think the key factors on this slide, which have not changed, is that the American Cancer Society is one of the only nationwide cancer-focused organizations that focuses on all cancers and is not a specific cancer focus like Susan G. Komen, for example, and that we do research, education, service, and advocacy. And in fact, our sister organization, the Cancer Action Network in Washington, D.C., our CEO is also the president of that, the cancer advocacy work. So one of the other reasons I really got excited about coming to ACS is it really offers that opportunity to help translate research. Quite often, I would say at least once or twice a month, myself or one of my colleagues gets an email from someone at ACS CAN that says, hey, we need to know something about e-cigarettes. What's the latest research showing? Where are we going? Because they really need to help understand what is our position and how are we going to help us support legislation on different exposures. So research at the American Cancer Society actually dates back to about, dating back to the early 1920s when the American Cancer Society leaders, the society started in 1913, began to notice this emerging epidemic of lung cancer in about the 1930s. And it was kind of going in parallel with this increasing consumption of cigarettes. And it was this increase that really led the society to kind of think about launching a research program to help answer the question of why. What is driving that epidemic of lung cancer primarily in men? And Mary Lasker gave a gift to the American Cancer Society that really helped launch both its extramural grants department as well as an intramural research department. So in 1947, Dr. Hammond was appointed as the head of a very small intramural epidemiology and surveillance department that was really launched to help kind of assemble better statistics. There were very, very few places in the country where there were good statistics on cancer rates being assembled, as well as to help answer some of these questions about the lung cancer epidemic and help understand why it is that these rates were increasing. It was the first study the first large prospective before I get into that, today the American Cancer Society is research programs are basically fall under Otis Brawley, who's our chief medical officer, and Liz Ward, who heads up the intramural department, and Bill Chambers, who heads up the extramural grants department. Intramural here, as shown here, is comprised of 5 programs. I head up one of them. Surveillance is right there, and you might all be familiar with Cancer Facts and Figures. We have a Statistics and Evaluation Center, and that's really a cool center because what they do is help the society understand how the programs we deliver out in the communities, are they effective or are they not? Are they actually costing us but not doing anything? And so the Statistics and Evaluation Center really helps understand how the ACS is delivering programs and whether or not programs should be sunsetted. We have a behavioral research center, which is headed up by Kevin Stein, and they have a couple of about multi-thousand person cohorts of cancer survivors, and they do a lot of work helping translate research to our mission delivery survivorship program. And then Jeff Drope heads up our economic and health policy program. It's a very large international program. A lot of their work focuses on tobacco, and they've helped understand cost effectiveness of tobacco control at an international level. So epidemiology, this is who we are currently. So we're comprised of about 9 doctoral level investigators. We're supported by a very, very talented group of study managers who are responsible for collecting the data and the biologic specimens and preparing initial data sets, which are then further annotated into analytic data sets by our statistical analysts, and who are responsible ultimately then for helping both us write papers and analyze the data, as well as oftentimes they also lead and write their own analyses. So we're about 32, 33 people right now. Our mission in epidemiology is to provide that scientific evidence that supports efforts aimed at reducing the cancer burden. And we do that primarily through large prospective studies, conducting these large nationwide prospective studies. We also play a role, as I mentioned, in communicating findings from those studies, both within the organization as well as at a national and international level. Our priorities are really research that focuses on the greatest impact on reducing suffering and death from cancer. So we focus a lot on the common cancers and risk factors, high impact unanswered questions, and I'll talk a little bit about some of those that my colleagues and I have both done, as well as high quality productive collaborations that allow us to extend our expertise. So going back to the first prospective cohort study, which was launched in 1952, and Dr. Hammond's wife actually came up with the idea of leveraging the Army of Women, the U.S. Army of Women, who helps support the American Cancer Society's programs and fundraising, to go out and recruit their husbands, their friends, into a cohort of about 188,000 men in 9 states, and to ask those men to fill out about a 4-page survey, and then follow those men to see if the men who reported smoking were more likely to die and what did they die of. And so their mechanism there, and shown here, are Drs. Hammond and Horn. And I should mention to you that both Drs. Hammond and Horn were smokers, and they didn't believe that smoking caused lung cancer. And in fact, you know, at the same time, there's a study being conducted that some of you might have read about called the British Doctors Study, being conducted in England, to also, Dahl and Pito were trying to basically disprove the same thing. Smoking does not cause lung cancer. All these case control studies that had shown this were flawed. So within just a few years, the research results were in, and the study convincingly showed that indeed smoking causes premature death from both all causes, cardiovascular disease, and lung cancer. And so lo and behold, they switched to pipes. But the truth is they actually did quit the pipe smoking as well. I think it soon became clear that it was more beyond just cigarette smoking. Since that time, we've launched 3 other large prospective studies. There's Cancer Prevention Study 1, which was launched in 1959. And this time they were wise enough to recruit women, but used the same mechanism of going door to door and asking people to fill out surveys, sending those surveys to the national office in New York City. And then every year, that same person who enrolled, the volunteer who enrolled participants, would literally go knock on the door of the person they enrolled. Are you alive? Are you dead? And if someone had died, they would then get some information, and the people in New York would request death certificates. And so Drs. Hammond and Horn and their colleagues would pour through not just the surveys, and you can imagine what this was like in the late 1950s. It wasn't like today where you just go online and fill out some surveys and everything's done magically. It was a lot of work. And they also had to get the death certificates and code these for cause of death as well. And they enrolled about 1 million women in CPS-1, and they, lo and behold, they showed that in CPS-1, smoking is also a cause of lung cancer in that cohort as well. And then in 1982, CPS-2 was launched, and we continued to follow the entire cohort for mortality. Our most recent linkage to the National Death Index was in 2000. Was for deaths through 2012, and over half the cohort has now died. We're about to update that for deaths through 2014, and we're anticipating an even more dramatic loss of life in that cohort. But this cohort has essentially been followed for a very long time, and I'm going to show you some really intriguing results from CPS-2. I'll talk a little bit about how we've enhanced the resources in CPS-2. So in CPS-1, it was essentially a one-time survey, essentially. In CPS-2, there's a sub-cohort that we also follow for cancer incidence, and we've been able to enrich that cohort quite extensively, the resources there. Our newest cohort, CPS3, we began enrollment in 2006. We completed it in December 2013. You'll see that there's been a drop in the number of people who've been enrolled in CPS3. So we went from 1.2 million down to 300,000. And that's because we have a blood sample on everybody enrolled in CPS3. I think of the 304,000, we didn't get a blood on maybe 4,000 or 5,000, and that's because they're the children of CPS2 who enrolled through a follow-up survey. We also have a waist circumference measurement that was enrolled, that was taken at the site when they enrolled. So it's a little bit of a different mechanism rather than having people go knock on doors. So with that said, our smoking work has contributed from these cohorts, has contributed not just, I feel like this slide is out of place, I'm sorry. I'm going to go to the next one. In our cohorts, our research has been kind of unique in that we've been able to actually demonstrate the impact of tobacco on mortality, both from lung cancer and all cancers, over 50 or 60 years. So if you look here, our work has essentially shown that in Hammond and Horn was published in about the 1950s, CPS1 in the 60s, and then CPS1 and Hammond and Horn contributed to the first Surgeon General's report, and then also to multiple other public health efforts to help reduce tobacco consumption in this country. That work overall has helped lead to the declines in lung cancer rates in both men and in women. But with that said, the death toll due to smoking overall continues to rise. And the society actually creates the relative risk estimates that the society from CPS2 that the CDC uses to then come up with a number of deaths due to smoking in this country. Eric Jacobs in my group, Michael Toon had been doing that work. Eric Jacobs just helped CDC with the 2014 Surgeon General's report. And you can see that the number of deaths due to smoking has gone up to about 480,000. But we quickly learned using another large collaborative data set that number is actually too low. And Brian Carter and Eric in our group actually published this study in New England Journal of Medicine about a year and a half or so ago, in which they showed that the number of deaths is actually about 15,000 low. $480,000 is actually 480,000 deaths is actually an underestimate. It's probably closer to about 495 or 500,000, and this number is likely going to continue to increase. And that's based again on some recent studies that have been done that have shown that COPD rates in women in particular are not necessarily coming down. And so there's a variety of work that we continue to do to contribute to what we know about tobacco and lung cancer. And so beyond etiology, I mentioned to you that we have a behavioral research center at the American Cancer Society. So recently we've kind of been reaching across the aisles, something that there was not a lot of that happening at ACS several years ago. And Lee Westmoss is a smoking cessation expert, and he worked with Eric and colleagues to look at the teachable moment of a cancer diagnosis. Is that a time in which people can be helped to reduce their tobacco consumption? And what they showed is that indeed the quit rate is considerably, two-year quit rate is considerably higher in people who are diagnosed with cancer than people who aren't. And this does not include the lung cancers. And the 4-year, even after 4 years, there continues to be a higher quit rate. So Lee feels like there's an opportunity here to develop smoking cessation programs for cancer patients now that we have scientific evidence to actually show that quit rates are higher. In addition, and I know I'm spending a lot of time on tobacco, but it remains the #1 killer of cancer in this country and worldwide. In our new cohort, CPS3, we uniquely asked, no other cohort has ever done this, we asked questions about quit attempts. How many times have you tried to quit, and in which ways did you try to quit? So we have some very nice detailed information on smoking cessation. And Vicki Stevens, who's a geneticist in our group, wanted to look at beyond nicotine dependence. Can we identify the genes that help contribute to smoking cessation? And so in CPS3, she did a small candidate gene study looking at about 80 genes that are somehow related to smoking in some way, shape, or form. And she divided a group of people who quit easily. They quit after their first attempt versus those people who reported it took them at least 10 or more times to quit, or they didn't quit. So they've attempted it at least 10 times. And she identified 11 SNPs, or genetic variants, that contribute to a genetic risk score, essentially, here. And what she found was, indeed, she could predict whether or not, using this candidate gene approach, she could predict whether or not people would be an easy quitter or a difficult quitter. This study has now been used to provide the pilot data needed for a large R01 that was just submitted to do a large GWAS to kind of look beyond just the candidate genes, but just now canvas. And the long-term hope here is that we can identify biologic pathways that may help in the development of smoking cessation aids. It also may help with risk prediction and helping people, predicting how easy it will be for people to quit smoking versus not. So as I mentioned before in the first slide that I should be after this one, our data have actually contributed to every Surgeon General's report on smoking and health. And the impact of that work does continue. And in fact, in CPS3, we now have data that we're collecting on e-cigarette use. But what about beyond tobacco? We know that there are many risk factors beyond tobacco for cancer. And in fact, the Cancer Prevention Study 1 provided the first epidemiologic evidence that obesity or excess body weight increases to premature mortality from both all causes as well as cancer in both men and in women. My predecessor, Jean Kell, published this landmark paper in the New England Journal of Medicine showing that obesity increases the risk of 10 separate cancer sites in both men and women. And these data have been used in multiple large national and international reports on the effects of obesity. Michael Toon has published published this paper showing that alcohol consumption, light alcohol consumption was associated with a reduced risk of cardiovascular disease, but increased light and heavy consumption was associated with an increased risk of several cancers. And as you know, this kind of J-shaped relationship between mortality, all-cause mortality, and especially coronary heart disease mortality, and alcohol consumption is what is kind of a paradigm shift for our guidelines in terms of the fact that the American Cancer Society, while we know alcohol is a carcinogen, it's difficult to recommend that people don't drink if there may be a cardioprotective effect. And I'm going to talk more about that towards the end when I talk about my own research, because I think that's being called into question. So beyond some of the survey-based data from the CPS1 and CPS2, over time, the resources in CPS2 have been enhanced. So shown here is the 1.2 million people in the baseline cohort. And then in 1992, a sub-cohort of about 184,000 men and women living in 21 states with high-quality tumor registries were asked to complete about a 20-page survey that included a block food frequency questionnaire. And so since 1997, we have been updating exposures in CPS2 every 2 years, and we ask our participants to self-report a cancer diagnosis, and then we go verify that diagnosis. Beginning in 1998 and through about 2000, we collected a blood sample from, we call it our LifeLink cohort. We collected our blood sample from about 40,000 people, and then from 2001 to 2003 or so, we collected a mouthwash sample in an additional 70,000. So we have a DNA sample on 110,000 of our CPS2 participants. This has really also proven to be an incredibly rich resource for a lot of collaborations, and I'll talk about that in a minute. Beginning in about 2010, we began both the retrospective and the prospective collection of of tissue samples for 4 cancer sites: colorectal, breast, hematologic malignancies, and prostate cancer. And we chose those 4 cancer sites, first of all, is because they're common, and because we have investigators who are scientifically very interested in those cancer sites. And we felt like people would be motivated to consent to allow us to get those tissue samples. We've been incredibly successful. We have breast tissue samples on over 1,500 of our CPS2 participants, over 1,000 colorectal samples, over 1,200 prostate tissue samples, and in heme, over 500 hematologic malignancy tissue samples, which has actually led to an R01 grant that was recently funded in collaboration with investigators at City of Hope. So these resources have really allowed us to do a lot more work. So as I mentioned, that 1992 survey had detailed information on diet. This is a study that was published using those data by Ann Chow, showing that red and processed meat intake increases the risk of colorectal cancer. And if you remember, about a year ago now, the IARC, the International Agency for Research on Cancer, called processed meat carcinogenic for colorectal cancer, and red meat a probable carcinogen. And it's these data and others that help contribute to that classification. We've continued our obesity-related research. This is just a number of projects I just want to point out, including using our biospecimen resources to look at biomarkers related to obesity. In total, our data contribute to the American Cancer Society's guidelines on nutrition and physical activity for cancer prevention. In 2012, our guidelines, the basic premise of our guidelines at that time were to adhere to and maintain a healthy body weight through adulthood, adopt a physically active lifestyle, consume a healthy diet based on plant foods, and if you drink alcohol, limit consumption. When I got ACS, I asked the question, how do we know our guidelines work? Has anybody actually ever evaluated these guidelines? You know, we put these guidelines together based on separate studies. But what if you put them all together? And so Margie McCullough, our strategic director for nutrition, created a score, essentially. And people were given a score of if you didn't follow any of our guidelines at all, you were given a score of 0. If you followed them a little bit, each component just a little bit, you were given a score of 1. If you followed our guidelines to the T, you were given a score of 2. So the total score could be 8. And you can see here for overall mortality, there was a beautiful dose-response inverse relationship between following the American Cancer Society's guidelines and premature mortality. For cancer mortality, it was statistically significant. It was just not as dose-related. When we started pulling pieces out of this, much of that reduction is due to weight maintenance, but not all. Each of the components actually contributes significantly. And so I think that this provides really nice evidence that actually following our guidelines may have benefit. And what's really nice is these these findings have been replicated in the American Association of Retired Persons cohort, as well as in the Women's Health Initiative, and I think in the PLCO, the prostate, lung, and colorectal and ovarian cancer screening trial. So it's really nice that not only did we see it, but there has been replication as well. In terms of utilizing our tissue specimens, Pete Campbell in my group is a colorectal cancer expert, and so what he's been doing is working with investigators at the University of Washington and the Fred Hutchinson Cancer Research Center to look to measure the 5 markers or so that identify the 5 molecular phenotypes of colorectal cancer based on their microsatellite instability or their methylation status. And they're beginning now to analyze this data and look at risk factors based on separate molecular phenotypes. And based on case control studies, there have been findings that indeed the risk factors differ. So the reason that's important is because for colorectal cancer, it was long argued that obesity may not be related to colorectal cancer risk in women. And that's because women develop usually a different kind of more often develop a different molecular phenotype than men. And so once you're able to kind of parse these out instead based on gender, but based on molecular characteristics, we do indeed see differences based on their molecular phenotype, which could in part explain that disparity or that difference between men and women. I mentioned that our scientific collaborations allow us to enhance our expertise. We're actually involved in greater than 35 active collaborations at any time. I would say on a weekly basis we're developing new contracts with people. Some were retiring, some were developing new ones, some were just extending the length. Together, these have allowed us to use tissue serum from more than 7,000 of our participants and DNA from well over more than 30,000 of our participants. And I'm really excited because we're on our way of actually getting genome-wide data on hopefully my goal is to get it on the entire 110,000. We're at about 25,000 right now, but we're trying to work with NCI and convince them that this would be a great thing to go in together. So I want to highlight a couple of these collaborations. This is probably one of our oldest, most long-term collaborations, and something, Miles, that you might be interested in, is that Arden Pope, who is actually an economist at Brigham Young University, published a paper back in 1995. And what he had done is he had taken air monitor data that had been collected in about 150 air monitors throughout the United States and looking at fine particulate matter exposure in the early 80s. And he related that to subsequent death from both all causes, COPD, lung cancer, and other causes in the CPS2 cohort. And though this finding, along with a paper that was published at the same time with the Harvard 5 Cities cohort, were used to help establish the guidelines for the Clean Air Acts of the 1990s. And they continue, this research does continue through a collaboration with a man named Dan Krusky up at the University of Ottawa and Michelle Turner. And these data, along with a lot of updating of the data, smoothing of the data, you know, they came in and geocoded our cohort, and they have used that to develop new exposure measures. But these findings are widely cited and continue to be widely cited in EPA recommendations, much to sometimes Congress's chagrin. It causes us a little trouble sometimes. But also were recently cited in the most recent IARC evaluation on PM 2.5 particulate matter exposure. So again, kind of talking about the impact of these studies on helping change policies and lead to new policies on reducing exposures to air pollutants. Lauren Terra's in my group here, who is the hematologic malignancy expert, recently published a paper looking at radon exposure. Working with Michelle, who had developed the exposure measure for us at the zip code level, and showed that indeed exposure to radon in the homes was associated with a higher risk of hematologic malignancies in women, and not to quite the same degree in men. We hypothesize that one reason may be, and at least in this cohort, in this age group, is women may have just been in the home a lot more than their counterpart, their husbands. Another kind of high-impact, high-value collaboration, which is led by Alpa Patel, is through the National Cancer Incidence Pooling Project of Physical Activity. And what she and her colleagues did was pooled data from 12 different studies that were comprised about 1.4 million individuals, and showed that indeed physical activity reduces the risk of death from at least 13 different cancer sites. And that was kind of a big deal this year, because previously it was kind of suggested for some of these cancers, but the power of putting together the data across multiple cohorts, and I think the reason that, you know, these cohorts were really attractive is they had all collected the data in a similar, the physical activity data in a similar way. So these data could all be pooled, put together, and again, show really nicely this beautiful reduction in risk for many cancer sites. I told you that we do a lot of work on collaborations on the genetics, and this is just one. And the Breast and Prostate Cancer Cohort Consortium, BPC3, was kind of what launched us into this effort. It was originally back in the early 2000s a candidate gene study primarily for genes related to hormone and growth factor metabolism. Since then, it's grown and gotten to be much larger, and it was only 4 cohorts at that time. It's grown into something much larger. And our data have now been contributed to either the identification or validation of every known genetic breast cancer genetic variant in the population. This is now being taken to the next level. So there's a project being done by our collaborator at the Mayo Clinic who's actually now sequencing all of the breast cancer cases and controls for about 35,000 cases and 35,000 controls. And the reason this is any of you know who Mary Claire King is? She is the person who originally, she and others, originally identified the BRCA1 gene, the region in BRCA1 that was associated with a very high risk, high penetrance breast cancer. And it's her feeling that we need to be, more women in the population need to be screened. Actually, it's her feeling all women in the population need to have their breast cancer genes sequenced. But we don't really have any good data on what are the population estimates of these variants. So that's one problem, and there's another problem. Oftentimes women are sequenced, they go to commercial laboratories, they get their gene sequenced, and they have a variant of what's called a variant of uncertain significance. It's not been seen before, it's not sure if this is pathogenic or not, and so there's a lot of uncertainty, and that causes a lot of anxiety for women. Some women choose to have prophylactic mastectomies, oophorectomies, going on chemotherapies, getting more frequent screening, and it's not sure that's the right thing for them. And so this new project is not only going to help us learn more about these variants in the population, but we're now then developing a collaboration with IBM Watson to feed all of that information to Watson as well as what's known in the in silico world to help us better understand, are these variants of uncertain significance important or aren't they? So our feeling is, again, the impact of this work could be quite tremendous ultimately, but you can't do that alone. None of our studies can do that by ourselves. We have to collaborate and work together. So again, extending not only our expertise, but if we get so far as to get this collaboration with Watson, it'll also extend our technology. So despite all this progress and all this good work that we've done, the fight continues. And now I'll just stop here and take any questions, but next I'm just going to give you an example of my own research. But if you have any questions about the cancer prevention studies in general, I'm happy to answer them. Yeah.
I'm curious as to, with the CSR2 that you did, to keep track of those patients that perhaps have mental illness as well, so they might be more likely to have addiction, say, properties, qualities, genes, genetics, and the question is, of those patients, do you know if they have a higher risk or a lower risk?
Yeah. So we do have some self-reported information, or sometimes it's a son or daughter that may report if the parent is in a nursing home or something. So we have some self-report on mental illness. We don't get a lot of detail because it's a cancer cohort. We have some information. We have some information on that. We have not mined it, though. So if you're interested in knowing, if you go on to the cancer.org website and you go into research, or else you can email me. Ultimately, all of our questionnaires are available online, and so you can search for our surveys and see what we've asked people. Okay,
I have a question about scientific sociology. So as I talk to people at other universities who are part of these large cohort consortia, what they're telling me is that they are now spending most of their time on the phone or in emails or otherwise communicating across the collaborative process. Is that what you're seeing as well?
Yeah, I mean, I would have never thought I would spend as much as time as I do with lawyers. And this year, I'm the chair of the NCI's Cohort. One of the things I organized was a work group that would focus on creating collaborative agreements that were more user-friendly, but unfortunately, each institution. And what we're finding is some institutions where investigator, and I'll use Cambridge, University of Cambridge in the UK. We'll do a process where we will vet a collaboration for 2 weeks. Collect everybody's feedback, then we're going to create one, and you either sign on or you don't sign on. But yeah, it's very difficult. And one of the ways we've taken a little bit of the pressure off of each investigator is I have a coordinator who now manages at least the internal contract. It's a lot of work.
Question about the variant that the investigator breast cancer or mutations that are known. I think we're talking mostly about BRCA mutation. That really surprised me, and I guess I'm wondering, are we talking about a specific population screening? And then I guess I wonder about other risk factors like nulliparity and having a baby. A lot more associated with breast cancer, at least in terms of the number having that risk factor. What are all these different risk factors prioritizing which ones?
I think it's a great question. So we have through all of the epidemiologic survey research, right, we've identified a number of modifiable and non-modifiable. Parity wherever you want in that. I think there isn't a full appreciation in the population of known risk factors even. Now we're going to, some women who are very interested, go get so I think that we have a hard job of trying to help educate the public. I don't say this lightly, but I say this. It shocks me every time, I've been studying this for over 20 years, the alcohol breast cancer link. It shocks me when people say, not read Ladies' Home Journal. I mean, every October there is something about this in a lot of the women's journals. So I think that we have a communication problem. The other problem is our work is done at a population level. Now trying to communicate individually. Just over the weekend, I was emailing back and forth with our communications folks at ACS, and they said to me, "So, Sue, in the WCR, the World Cancer Research Fund, American Institute of Cancer Research, it says that 20% of all cancers can be attributed to the 4 factors, maintaining healthy weight, et cetera. Does that mean that you can reduce your risk, a woman can reduce her risk by 20% if she does these things?" We have a long ways to go, and these are people who are embedded in the cancer world. So I think there's a lot of work of trying to figure out. But if you're interested in knowing more about risk prediction, this is a plug for my own. We have the Molecular Epidemiology group of AACR has a meeting coming up in Orlando next month, where worldwide we have a bunch of people coming together to talk about risk prediction.
My question is may not really a question. I'm interested in how lawyer is involved in your work and how you work with the lawyer.
That's a loaded question. So every time we collaborate with external, we give our data or our biospecimens to someone. That through a contract. Only all the human subjects issues are addressed. And there is an institutionally signed contract by their institution that talks about the protection of our data. And so we work with the lawyers to confirm the language in contracts, because sometimes we initiate the contract, but Harvard University X's out many parts of it. And sometimes Harvard will, or I'm using that as an example, will initiate the contract, and we have to add into it. So I don't write that language. I'm not a lawyer. I don't speak lawyer talk. The piece, there's always an attachment. Talk about the research, but there's a lot of language around. That's just one example.
The first answer to that is I'll refer that to my lawyer for an answer.
So a lot of the work is that modalities and looking at when do people start screening. So one of the areas that I think has been fascinating is colorectal cancer, of course, in the 50s and younger situations. How much, and I'm sorry I was late for your presentation, how much work are you guys doing in concert and others that are looking at modeling studies? I think we see two different ideas that ultimately randomized control trials in some of these areas long, whereas modeling studies might be the way to go, but there's a lot of criticism and critique. So I'm just wondering, given the political nature of the underpinning, I think, where are you guys with that, and what's your kind of general?
So in terms of the epidemiology program, we are largely etiology. Okay. But we do not get involved in the prevention. I don't know if Tim, you want to.
Yeah, so you mean in terms of screening policy and screening recommendations?
So I was just wondering if any of these really linked into some of the modeling from a very more prevention-based.
So there are 5 programs in intramural research. One of the programs is a health policy program, and 90% of their work has historically focused on tobacco. They are beginning to get more involved in screening, but they're just starting. That group is, yeah. Not as much, yep.
With the breast cancer together, and this whole idea of genetic risk factors, is there any generational or an effort to study the cases in a generational kind of family history? Probably your best bet in terms of the risk. So instead of maybe doing, there's focus on genetics, but then maybe you focus on this generational, since you have the cohort, or at least the cases in the future, generational factors, and because you have them, you can ask them questions about what they're doing in their lives and kind of.
So you're thinking the kids of CPS2 type of thing? Yeah. So if we think about, so I'm going to try to answer your question 2 ways. If you're asking, do people with a family history, do they behave differently than people without a family history? Sometimes that is true. We just finished a coffee paper. I just finished a coffee paper, and remarkably, and I don't know why this is true, people with a family history are more likely to drink coffee. Don't ask me why. And so you know, I think there may be screening behaviors that may differ because of your family history. Our women in CPS2 were enrolled when they were in their 50s and 60s. So back in the 80s and 90s. So we didn't have quite the same appreciation then for what we have now. In our newest cohort, CPS3, we only have about 7,000 total men and women participants who are kids of CPS2. So there's not a whole lot we can do there. With CPS3, our newest cohort, people enroll in CPS3. First of all, people enroll in American Cancer Society cohort studies because they are passionate about cancer. They already are passionate about this. That's why they are supportive of our work. They are different. We have a very educated, highly educated cohort relative to the general population. Our women have a much higher prevalence of family history than what you may see in NHANES data. And again, I think that's largely because we're a cancer-focused cohort. So I think it would be an interesting question to ask, you know, about changes in behavior as we may learn about, because we will always ask every 3 years when we resurvey the CPS3 cohort, we'll always ask new updated information on family history. You know, in the last 3 years, has your mother, daughter, sister been diagnosed? So we can start looking at changes in behavior without coming out and asking them, did that cause you to change your behavior?
So you have your questionnaires and things online, but you also have information about what has been collected when and what's available for use if that's always the tricky one.
We have, no. The short answer there is no. We have a policies and procedures for data sharing that's available online, so you can go in and read about it. I would say 90% of our requests come from our friends and colleagues that already know us and know what we have. And that 10%, some of these people we would never collaborate with. They're kind of crazy. Some of these people are new people that, you know, we've never talked to them before, but like I got an email about 6 months ago from, let me back up. Back in, I think, the early 90s, Michael Toon had published a paper showing that living at a certain latitude and longitude was associated with a higher prevalence of kidney stones. I think it was based on zip code. And so a nephrologist recently contacted me and asked, they want to reanalyze those data, except this time using our geocoded data. They want to actually use air temperature, NASA air temperature data, and to look at whether or not they can replicate that instead of just being based on lunge, but also on air temperature. And we thought that's kind of a cool use of our data. Sure, it's unusual, it's different. You know, as you know, they have to come to ACS to do the linkage to get the exposure information because we don't release geocodes. But yeah, so it's kind of best to just contact one of us and find out.
Crazy people mostly at Cambridge and Harvard? No.
What research are you doing now?
So you mean, what resource am I personally?
Yeah, you said you had your share.
Oh, this is, so an old passion of mine has always been pancreatic cancer. And it is one of the most common causes of cancer mortality in men and in women. In 1988, when IARC, do you all know what IARC is? Maybe, maybe not. Okay, raise your hand if you know what IARC is. Okay, thank you. I will explain it. The International Agency for Research on Cancer is a part of the World Health Organization, and back in the 1970s or so, they were mandated with evaluating the carcinogenicity of primarily occupational chemical exposures. But over time, they were asked to start evaluating the carcinogenicity of a whole lot of other types of exposures as well. So they've developed what's called the Monographs Program, which is essentially they bring in a world panel of experts, people from all over the world representing all kinds of areas to evaluate a topic. And so in 1988 was the first time they evaluated alcoholic beverage consumption. They had, demographers, to look at consumption patterns around the world. They had epidemiologists to look at what do we know about associations. They had animal modelers. to look at, if you feed animals alcohol, what's happened, and then to try to understand mechanisms. And what they found that was, indeed, there was evidence at that time that it's a cause of like aerodigestive and liver cancer. But at that time of the studies that had looked at pancreatic cancer, they said it's unlikely to be causal. But in 2010 monograph, this was the committee I actually participated in, for the cancers, the pancreas, we felt like the data were sparse because or inconsistent. There were very few prospective studies. And then in 2012, kind of a reevaluation of a whole lot of things, they came out and said the possibility that residual confounding by smoking may partly explain any association. But the real issue isn't necessarily just is alcohol a cause. We know alcoholic pancreatitis has been linked to alcohol, or to pancreatic cancer. So is it really heavy consumption? And so we set out to try to help address this issue, this limitation that IARC had pointed out, and to examine both the dose-response relationship and to better understand this confounding by smoking, a known pancreatic carcinogen. So we had an analytic, we took our CPS2 baseline cohort of 1.2 million men and women. We excluded the people with prevalent cancer, poor alcohol data, poor smoking data, ended up with about 1 million people. People had, in 1982, had filled out this beverage grid, and so we could come up with drinks per day of the 3 different types of beverages, beer, wine, or liquor. And we could identify both the non-drinkers, be they either never or former, and less than daily and daily. I can tell you right now the formers didn't look any different than the nevers, so we just combined them. We had vital status. I won't go into how that's ascertained, but essentially at the end of the day, we identified about 6,000, more than about 6,800 pancreatic cancer deaths in this cohort. By far the largest, more than 4 times larger than any other study that had been published to date on this topic. And what we were able to do is look at the dose response. So first of all, we just adjusted for, without adjustment for smoking, we looked at the dose response. You see evidence of an increasing risk at about 2 drinks per day. But when you adjust for smoking, the increased risk is more evident at about 3 drinks per day. And results were generally similar after excluding the first 5 years of follow-up because of potential for reverse causality. But then we wanted to look at the stratified results. So what about in the never smokers, the lifelong never smokers? And indeed, you do see an increased risk, but you don't see it until at least 3 or more drinks per day. So this suggests that there is an association, that it is with the heavier amounts of consumption if 3 drinks is heavy, and that, it isn't due to just any observation that we've seen in the past. It wasn't due to just residual confounding by smoking. I will say to you that one of the things to think about when it comes to alcohol is people notoriously underreport their alcohol consumption. It's about 50% of the the alcohol disappearance data goes unreported in surveys. So is this really 3 drinks per day or is it really 6 drinks per day? I don't know. And there's probably a relative difference in reporting. Like people who drink 1 drink probably don't mind telling you I drink 1 drink. People who drink 6 drinks probably are more likely to say, maybe I don't want to admit to that. Maybe I'll admit 4. It's a little harder. And then, you have your Lysol drinkers to get their alcohol. So we don't record that at all. Anyway, so I feel like our results do contribute to these sorts of international recommendations and guidelines and in terms of, in this case, the carcinogenicity of a cancer that has not yet been linked, confirmed to be linked to alcohol. So at the end, the society's cohorts are unique because of their size, their sequential coverage over 60 years. The yield from the collaborative research has just been huge. The research emphasis on unanswered questions. We contribute both to work at the society as well as nationally and internationally. And this work really has helped contribute to policies, both in the U.S. and worldwide. Like I said, the air pollution findings alone are widely cited in the EPA documents as well as in our own, as in the WHO documents. That said, I'm happy to answer any more questions. Oh, my last plug. Sorry. So we, just in case you think we just get handed money to play with, we actually go through peer review every 5 years. An external group of people come in and evaluate our work, evaluate our progress. Our last one was in March of 2014, and the committee said, you guys are bogged down in process and contracts and things like that. You know, a postdoc program could help accelerate your productivity. And they encouraged the organization to find a donor, essentially, or a sponsor for a postdoc program. And so I wrote, like right away I got on it, wrote a proposal up, gave it to our major gifts people who go out and find money. And they were able to find a donor who is willing to pay, the entire program, 5 postdocs. Each postdoc gets about 3 years. It's a quite generous salary relative to the T32s. And they get money for a biospecimen-based project as well. Anyway, so a donor is willing to put up half the money, but it's a matching grant. So ACS has to go raise the other half in order to get half. But they're doing it on a postdoc by postdoc basis. So we have enough money. We are launching this. Probably this week we'll be able to put the ad out there. And we have our first 2 postdocs that we can recruit, and we're well on our way for the funding for the 3rd one. Anyway, so if you have any great students, we will be looking for people to come work with us and help use our resources. There was a question.
About your research, I thought it was fascinating. Question yet, perhaps, but did you determine if there's certain beer versus?
Yeah, yes, I did. I just didn't have a long time, so I didn't put all those slides in. I just wanted to give you a flavor for how we address unanswered questions. The association is strongest for liquor consumption, and that's not surprising to me. Couple reasons why. People can tell you very well how many cans of beer they have, how many glasses of wine. Even though people pour different sizes, it's usually within the 4 to 6 ounce range. You know, you're not getting a 12-ounce glass of wine. You may drink 12 ounces in, an evening, but it's not a glass, and we usually ask by glass. Liquor, totally different ball of wax. There have been studies that have gone to shopping malls, and they've asked people to pour what they think is a typical amount of alcohol that's, served to them in a restaurant. Some people just keep pouring and thinking, geez, that maybe is what they're consuming at home, where some people kind of get it right. But it's really difficult. And so what we're thinking is people who are reporting 1 or 2 drinks of gin and tonic are actually drinking. The amount of alcohol in there is probably more closer to about a 3 or 4 drink. And so when we think about this dose response issue and the fact that, we know alcoholic pancreatitis is linked to pancreatic cancer, it probably is a heavier consumption. And that's why we're picking it up better with the liquor. All right.
If there are any other questions, then you can go and corner Dr. Jeffrey, but we meet her at about 1:15 or 1:20 to head back.
So one thing that, just to let you know, and those of you who are students, that you keep learning. I was challenged last week by my colleague Margie McCullough because, and Tim's going to appreciate this, I mentioned to you that there's this question of whether or not drinking alcohol, even at low amounts, 1 to 2 drinks per day, is cardioprotective, has been called into question through Mendelian randomization studies. I really am not completely sure I completely understand Mendelian randomization studies, because when I see it, I see, things like 3% of obesity can be explained by the known obesity-related genes. But they get excited about that, right? So this has happened with alcohol, and that it actually caused the UK to change their recommendation for consumption. Mendelian randomization studies suggesting that maybe alcohol is not cardioprotective. So I'm going to become our local expert because I'm committed to really diving into this from the alcohol perspective about this Mendelian randomization issue. I think it's an important message. It's part of our message. Does ACS need to change its guideline on this? Thank you.
So I work with Betsy and Tim very closely. Still trying to think about all of these large modeling studies that take all the data, but then yet have the predictive value of all the epi, and how does all this stuff start to coincide? A quasi-population level, start to look at more generalizations about people who fit into certain contextual elements.
So come to this risk prediction conference in Orlando. Some of the leaders in the world are going to be at this talking about this.